Swayam Prabha. Digital India. Educated India.
We finished the light reactions, and at the end we understood that at the end of light reaction the products are reduced equivalents in the form of NADPH, and then the proton gradient which leads to the formation of ATP. So now we are going to look at how this is used to fix the carbon dioxide, which is inorganic form of carbon, into the carbohydrates. Okay, so basically how are we making carbohydrates from carbon dioxide that is in the air? So this was figured out by Melvin Calvin, and then Benson and Bosch, and three of them. So won Nobel Prize in 1961. So this has three steps, so three stages. The first one is the fixation, like carbon dioxide getting incorporated into an already existing pentose, ribulose 1,5-bisphosphate. Ribulose indicates the presence of keto group like fructose. And the next one is the resulting molecules. The molecule will be 3-phosphoglycerate for every one of ribulose bisphosphate, which is 5 carbon, and when you add one more you have got 6, so you make 2 for trioses, and this is in the carboxylic acid form, and the next stage is the reduction of that to the aldehyde form, that is glyceraldehyde 3-phosphate. So these are actually the reactions of glycolysis, very similar enzymes, so this will not be difficult for us. We just need to place this in the overall context of understanding flow of carbon in the biological system. And the third is the regeneration of the entry molecule, which is the ribulose 1,5-bisphosphate. So to balance the stoichiometry, yesterday I told you the cycle starts with 3 molecules of this, and therefore 3 carbon dioxide. So you end up getting 6-phosphoglycerate, and after reduction 6 glyceraldehyde 3-phosphate. Out of the 6, 1 is the net gain, balancing the 3 carbons entered in the form of carbon dioxide. And the other 5 are converted into 3 5-carbon sugar, this ribulose 1,5-bisphosphate, that is the entry. And that regeneration requires ATP. Okay, for every molecule of ribulose produced, you have 1 ATP hydrolyzed. Therefore, for 3 of them, 3 ATP. So today we are going to spend a significant amount of the time focusing on this stage 1 fixation. So this entire cycle is called the Calvin cycle in honor of the scientist who elucidated this pathway. So this is the stage 1, ribulose 1,5-bisphosphate becoming 3-phosphoglycerate. So this is catalyzed by an enzyme called ribulose 1,5-bisphosphate carboxylase oxygenase, because it does both the reactions. And we simply call it as Rubisco. Okay, most of the people call this enzyme Rubisco. So if you refer to it by Rubisco, I'll be happy enough. But this is not that complicated. It's a long name, but it's very easy. It carboxylates this because carbon dioxide is fixed, and it is actually an oxygenation reaction as well, so it is oxygenase. And this enzyme has a very low turnover. Okay, only 3 molecules of carbon dioxide. If you compare this with enzymes like peroxidase, catalase, etc., they are like several 100,000 molecules per second they catalyze, whereas here the turnover number is low. And as a result, the chlorophyll has large number of rubisco molecules to enable sufficient carbon dioxide fixation. So due to this, rubisco is the most abundant enzyme in the biological system. Okay, so enzymes don't exist anywhere anyway. So rubisco is the most abundant of all enzymes. Like cellulose is the most abundant carbon material on earth. So we look at how this enzyme does the catalysis. So this is this enzyme structure. So you are only seeing couple of views. So you have a top view where you are seeing the 4 blue colored structures and then this gray. So these are the different subunits, 4 large subunits, the blue colored ones, and 4 small subunits that are the gray colored one. Okay, and the active site residues are colored yellow and visible through this ribbon model. And from the side, when you look at it, this is the view you get. Okay, so this has 8 large subunits. From one side, you are seeing only 4, and 8 small subunits. So the other 4 subunits are below this. You can see in the side view. So you see 2 here, and the other 2 are on the other side. And similarly, below, which is not seen here, 2 more. And the other 2 are on the other side. So 8 large subunits and 8 small subunits. It's a humongous enzyme, 550,000 molecular weight. And you see magnesium green here, and that's the main component in the catalysis. So we will see that as we go along. So I was emphasizing during our journey along with the electrons the importance of the vitamins. We saw niacin, we saw flavin, then we saw thiamine, we saw pantothenic acid, we saw biotin. All that we saw along the way as part of enzyme catalysis. Okay, and equally importantly, metal ions play a role. So we have seen heme moiety in cytochromes, and there you have ion. Then in the 4th complex in mitochondria, the cytochrome C, NAD oxidoreductase enzyme. So there we have cytochrome A3, and they have copper cofactor. So we have seen ion, we have seen copper. Then we saw manganese in water splitting complex yesterday, where the stable multiple oxidation states of manganese allows acceptance of 1 electron at a time, 4 electrons comfortably, and then donating 1 electron at a time, 4 electrons. Okay, so we saw manganese, and today we are going to see the importance of magnesium in the catalysis of rubisco. So all these, we call them from a nutritional point micronutrients. Okay, carbohydrates, lipids, proteins being the macronutrients. So these micronutrients, you will ensure sufficient intake when you have mixed varieties of vegetables, plant food in your diet. Okay, so that is why you should not rely only on French fries or pizza or burger as the only source of food. You have to eat a variety of food. Normally our traditional meal, whether South Indian or North Indian, ensures you have a variety of vegetables going in all 3 meals, and that ensures you have all of these. So this is a simpler version of the same enzyme found in bacterium. Okay, Rhodospirillum rubrum here shown. So this is the ribbon model, where it's very similar as if you have only a dimer here. Okay, so the blue 2 of them and gray 2 of them. With so you have the magnesium here, And this is an important lysine residue that plays a role, and this is the ribulose 1,5-bisphosphate substrate, the yellow color one. So this is a lysine that is in carbamoyl form in the side chain of the enzyme that plays an important role in the catalysis. So all of that coming in the next couple of slides. All right. So here a close-up of this region, okay, but particularly focusing on the magnesium. So here you see magnesium coordinated with atoms, actually oxygens from the substrate. Here 2 bonds in the substrate, one more with another substrate. Normally it is carbon dioxide in the actual catalysis. Here for crystallization they have used water. And then this is again from the enzyme side chain. You have a glutamic acid and aspartic acid. And this is the carbamoyl lysine, this red one. Okay. So this is in an octahedral complex forming 6 coordinations with the side chains and the substrates. Okay, 3 with the side chains and 3 with the substrates. So this is the active site structure of Rubisco. And this shows the reaction mechanism. So here, this as usual, this blue shaded area is the active site of the protein, the enzyme here. Here are those residues. And this histidine we didn't see in the previous one, because the previous one is only focused on how magnesium is coordinated. But for catalysis, this nitrogen becomes important, and that is why that is shown here. And this is the carbamoyl side chain. You know, carbamoyl is NH3COOH is carbamic acid. Here the lysine side chain NH2 is in carbamoyl form with this carboxylic group. And so this one abstracting a proton from the substrate. So this is the ribulose bisphosphate, and abstracting a substrate from this, again pay attention to the carbonyl group. You know, that is why it is ribulose that is functioning here and not ribose. So this carbonyl group, the chemistry of carbonyl group that we learned early on, so those are all core principles. You know, those chapters, at least that one particular chapter where we considered the 5 different kinds of reactions that we repeatedly encounter in biology, and some of the cofactor reactions like thiamine pyrophosphate, how it functions, lipoic acid, how it functions. So those kind of things you should revise multiple times and remember them. They are basically the core of the biochemistry concepts that you are learning. So this is you know, helping this to easily lose proton. Remember the carbon adjacent to carbonyl group, due to electron delocalization and resonance stabilization here, becomes carbanion. So this is more acidic than a carbon like this carbon. So therefore it readily loses the proton, and this shifts there, and this becomes, this takes that proton and it becomes a hydroxyl group. So you have one hydroxyl group here and another hydroxyl group that's going to form here, and a double bond here, a typical enediol structure. So that is what you are seeing here. So the abstracted proton is here, one, and then here you have the other one, and you have the double bond. So this is the enediol intermediate. So that allows carbon dioxide to be coordinated with the magnesium here. And now you see this acts as a base, a general base catalysis, and that aids in the electron transfer in these forms. And so this double bond is where this is broken, and this carbon is going to attach to this particular carbon. Okay, and the double bond is going to shift to this side, and that leads to a keto acid intermediate. Okay, so this is This is taken as the alpha and this is the keto acid. And there you have this carboxyl group attached, and that is why it is acid. Okay, keto acid formation. So this carbonyl group enabled that, okay, to initiate this reaction. Of course, magnesium and those coordination and this nitrogen, all of that matters. But in the substrate molecule, this carbonyl group is the crucial to initiate all of this. And now hydration of this double bond, shifts this bond out, and then you have this cleavage happening, releasing the first phosphoglycerate. Okay, so this is the hydrated version. So the double bond is reduced to OH, and then here you have the other OH. Then shifting of these bonds enabled by these linkages leads to cleavage of the bond here, and this becomes double bond. So this becomes a carboxyl group, so therefore it is phosphoglycerate that is released. And the remaining second one, the proton originally abstracted from this lysine, but now another lysine amino group provides the proton, and it's stabilized and released as the next 3-phosphoglycerate. So this is the reaction mechanism of Rubisco. Okay, the most abundant enzyme, and the enzyme that makes all our food. Okay, so this is how our food actually starts its journey from air, carbon dioxide. This is the way it ends up becoming 3-phosphoglycerate, and from there it is, as they say, rest is history. You already know what can happen to 3-phosphoglycerate. Reverse reaction that we saw in glycolysis will be glyceraldehyde 3-phosphate, and glyceraldehyde 3-phosphate by isomerization can become dihydroxyacetone phosphate, and if glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, if they join, they can become fructose 1,6 phosphate, and then you all the way go back to glucose. So those are ready for us to understand. So this formation is what is new and important here. So this is how carbon dioxide enters into the system. So although I said no more mechanisms, but this is one important mechanism. So please pay attention and understand this and remember this well. All right. So one extra detail with respect to Rubisco. So normally Rubisco remains completely in an inactive form. Okay. So this is activated by an enzyme called Rubisco activase. So this is inactive primarily because it is tightly bound with the substrate, and that prevents carbamylation of this amino group of the lysine. And in the presence of ATP, Rubisco activase expels this out and allows the binding of carbon dioxide and magnesium to form the carbamyl lysine here. Okay, so this is tightly regulated by light and availability of ATP and NADPH, etc. So now we go to the second stage, where it is the reduction of the phosphoglycerate to glyceraldehyde 3-phosphate. So that you can see here. Again, I'll try to blow it up. Yeah, so remember, this whole thing is happening in the stroma of the chloroplast. So this light green background is the grana, each one having one thylakoid flattened discs stacked up. So this is exactly reversal of what we saw in glycolysis. And again, the dehydrogenation step in the opposite direction as we saw in glycolysis produces glyceraldehyde 3-phosphate. And similar enzyme but present in chloroplast, isomerase, triose isomerase, produces dihydroxyacetone phosphate by rearranging bonds in glyceraldehyde 3-phosphate. And two of them can combine and form fructose 1,6-bisphosphate. Again, same steps in the reverse as in glycolysis. And this is a new enzyme, fructose 1,6-bisphosphatase. This is not there in glycolysis because there it is the opposite direction where you have a kinase phosphorylating it, PFK, phosphofructokinase 1. So this is the opposite of that, fructose 6-phosphate. Then isomerization to glucose, and then you have starch. And finally, this gets filled with starch, and as I told you yesterday, then it will become amyloplasts. Okay, chloroplast becomes amyloplast when it fills with a lot of starch. So this is one fate for the carbon dioxide that entered this route. And another one is it may be transported out via this inorganic phosphate triose phosphate antiporter, and the dihydroxyacetone phosphate that comes out, again isomerization to glycerolate 3-phosphate, and this might get into glycolysis to produce ATP. This is what happens in the leaves to generate energy for the leaves function itself. But at the same time, these are combined to form the fructose bisphosphate and to form sucrose. Okay, fructose, then isomerization to glucose, then glucose fructose disaccharide is sucrose. And this sucrose is the form in which carbohydrate is transported in plants from the photosynthetic organs such as leaves to other parts of the plant body. So the transport form of carbohydrate is sucrose. Okay, so that is why the stem saps are sweet. If you take grass and take the internodal region, the stem of the grass, and if you chew it, you will find it is sweet. That is primarily because sucrose produced in the leaves is being transported to roots, stems, and other parts of the plants. So remember, the transport version of the carbon dioxide fixed by the chloroplast is sucrose. And the long-term storage is starch, which is stored within the chloroplast, and the chloroplast becomes amyloplast. And to sustain the activities of the leaves, the glycerolate 3-phosphate, which came out as dihydroxyacetone phosphate, enters into glycolysis and TCA cycle to produce energy. So this is the third step of Calvin cycle. Sorry, this is the second step, the reduction of the glycerate to glyceraldehyde. Third step is actually to regenerate. So that is coming in the next one. Yeah. So this one again, I'll make big and then we'll go step by step. This will look complex, but it's actually not complex. All already familiar ones, steps. So the idea is we ended up producing glyceraldehyde 3-phosphate. Okay, so let me go back to the diagram and come back to this. Yeah. So we understand 3 carbon dioxide fixed into 3 ribulose bisphosphate producing 6 glyceraldehyde 3-phosphate. So we have net gained 1 glyceraldehyde 3-phosphate, and the remaining 5 must combine 3 carbon, like trioses must combine to make this pentose. And how is that done is what is our current slide's focus. This one. So here the main enzymes are these trans-aldolases and trans-ketolase. Okay. So the trans-ketolase is an important enzyme whose mechanism we are going to have a look at in the next slide. Quite simple but important. So here you have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate 3 carbon indicated by this, making the 6 carbon fructose bisphosphate. And the phosphatase removes 1 phosphate, giving you the fructose 6-phosphate. This is a irreversible reaction, strongly exergonic due to this hydrolysis of the phosphate group, and therefore it goes only in this direction. Then from here So this is a keto group, and 2 carbons above the keto group is transferred to an aldose. This is glyceraldehyde is an aldehyde one, and that is why it is called transketolose, ketolase. Okay, the enzyme, and the product is so this aldose becoming ketose, and then it produces the remaining part of this is the aldose again. Okay, it translocates the keto group, and therefore it is called transketolase. So you have 6 plus 3. So you have got 9. Out of the 9, you make 1 4 and 1 5. So the numbers are correct. That is the third step. Transaldolase. Here the aldehyde group is transferred, converting this into ketose. And then the second step is removing the phosphate. Third step is again the transfer of the carbons from a ketose to an aldose. And the fourth step is another dihydroxyacetone phosphate combining with this 4-carbon erythrose phosphate, aldolase reaction, the same enzyme as that, making the 7-carbon unique molecule that you will not encounter outside chloroplast. Okay, and this enzyme pseudoheptulose 1,7-bisphosphatase is unique to chloroplasts, and they are going to, this is again like this, an exergonic irreversible reaction making pseudoheptulose 7-phosphate, and this molecule via another ketolase reaction, transketolase reaction, then one more glyceraldehyde phosphate entering. First one we saw, then second one, and third one here. And that produces 2 pentoses, one aldose and one aldolose, keto sugar. Okay. And these molecules isomerize, lose a gain a phosphate to become ribulose 1,5-bisphosphate. For example, here this xylulose bisphosphate, 5-phosphate by an epimerase becomes ribulose, and that gets phosphorylated again, seriously endergonic reaction requiring ATP hydrolysis, unidirectional again. So the unidirectional ones are shown in blue, forming the ribulose 1,5-bisphosphate. And a very similar same reaction, an epimerase and this kinase reaction converts the xylulose formed here to this. These are identical molecules at the same steps. So now you almost see a cycle here. And this ribose bisphosphate isomerization to ribulose, aldose becomes aldolose, and the kinase makes a ribulose bisphosphate. So this is 9. So the numbers are shown, same numbers are shown when the reactions are the same. So similarly 8 here. Okay. So you have 1, 2, 3, 4, 5, 6, 7 and 8, and the 9th step ends up producing ribulose bisphosphate. So here I'm not going to ask for structures. The slide itself doesn't have the structures, but you need to have a kind of the understanding of the stoichiometry of how the carbons are jumbled by taking 2 3-carbon sugars initially and then adding 1. So essentially 3 trioses going through this series of cyclical reactions end up producing this pentose sugar. And this pentose sugar now has been regenerated. It can go for the stage 1, which is carbon fixation. All right. So now we will focus a little bit on the transketolase enzyme, because this enzyme we will not probably study too much biochemistry encounter elsewhere, but this is an important group transfer reaction. So therefore I want to go through it in some detail, not great detail, but some detail. So this molecule is very familiar to us. We have spent a lot of time with this thiazolium group and this carbon being acidic and can readily become carbanion. So we saw this in pyruvate dehydrogenase complex, where it carries the pyruvate's two carbons as hydroxyethyl form of the acetaldehyde group from the pyruvate. Very similar thing it does here. The two carbon groups of keto sugars it takes up. So here you have the reaction where you have the entry of pseudoheptulose 7-phosphate, the ketos. We are looking at that particular step. Okay, we are essentially looking at this transketolase. Okay, so this is our substrate here, and this is the other substrate. So you have the glyceraldehyde 3-phosphate. So this will come later. So first let us begin with this. So pseudoheptulose 7-phosphate, from that these two are going to be carried on this carbon. So here it is not hydroxyethyl because you have two hydroxyl groups in this. And so this double bond, this carbon is what is added to the thiazolium carbon. Again, this is aided by this carbonyl group. So now from here, so see, we are really not looking into the mechanism. We are only getting an understanding of just the major transfers only. So this bond is cleaved into an aldehyde, and then this is as an anion carried, and this is stabilized by this thiazolium electron sink. Remember the concept electron sink that we have learned. The conjugated double bonded ring structures help in temporarily juggling the electron among themselves and stabilizing these kind of unstable structures. And this now then is transferred to glyceraldehyde 3-phosphate, forming xylulose 5-phosphate. So essentially this keto group is now transferred to an aldose sugar, making that into a keto sugar. So that is why it is called transketolase. So this is how this transketolase transfers these two carbons to glyceraldehyde, making xylulose 5-phosphate. So this is the importance of thiamine pyrophosphate. Thiamine, remember, is one of the B complex vitamins. Its deficiency causes beriberi, and which is fatal. So that is this reaction. All right, next is budget calculation. Okay, so which I actually did it in the very first cycle itself. What is the stoichiometric relationship here? So 3 carbons with 3 ribulose phosphate. So the numbers are highlighted here, and this is the net gain. 5 of them we saw at that large chart, how they end up becoming ribulose 1,5-phosphate. So this budget calculation includes the ATPs that are produced or consumed. So here you see the 6 ATPs consumed, and then 6 NADPH, reducing equivalent. These are all equivalent of consuming food. Because we are doing a synthetic reaction. This is anabolic reaction. We are not in catabolism anymore. So therefore, the energy produced, remember, this was produced by the proton gradient created in the thylakoid membrane. And this you remember at the end of the photosystem, when the electrons are transferred to NADP, and that is reduced to NADPH. So that's how we have these inputs going in, and this is the net output. Okay, so this is the summary of photosynthesis and carbon assimilation reactions. So there are a lot of small sub branches and special river pathways and shunts, etc., which we are not getting into because we do not have that kind of a time in a one course biochemistry. in a 4-year program. And if you are very much fascinated by this, I would advise you to read the other portions in Leninger, the ones that I'm not covering. Okay, so that brings us to the conclusion of our discussions of how carbon, hydrogen, oxygen, do their dances in the living system. Okay, this energy for this dance is coming from sun. So as long as sun's energy falls down on the earth, you have these molecules dancing, transporting electron continuously in a cyclical fashion. Okay, so from water electrons are taken and the oxygen is evolved, and then later those oxygens the electrons flowing through the carbohydrate molecules and finally come to mitochondria and reduce oxygen into water again. Okay, so that is the electron circuit. And to make the electrons flow this way, the sun's energy makes the carbon, hydrogen, oxygen molecules to keep arranging, rearranging in the manner in which we saw. Okay, so this is the core path we have seen. We have not looked at the side branches. That is how I would like to view fatty acid biosynthesis and catabolism also. So if we have time, we will get into fatty acids. Otherwise, the core of biochemistry you have learned through whatever we have learned so far. Except that we have focused only on 3 elements so far: carbon, hydrogen, oxygen. Now we are going to the 4th important molecule. So in the order of abundance also, this is the 4th one, which is nitrogen. Okay. So you would have seen this plant. This is groundnut. Okay. If you follow American English, this is peanuts. So in India, we call groundnut. This is the groundnut plant. And this is the close-up of its leaves. Flower. So this shaped flower is a Fabaceae family member or legumes. And these plants are absolutely, pivotal for the nitrogen metabolism among the living organisms that we have. So we will see that one step at a time. So this is the aerial view of the plant, and from the branches, it has root-like structures going into the soil, and in the soil you have the seeds. Okay, the pod, and if you open the pod, you will find it mostly 2 seeds. Occasionally you may have 1-seeded one or 3-seeded ones, but mostly 2-seeded ones. And what is special about them, we will see as we go along. And this is one example. So this is an oil seed, and here you go, more of them. Variety of pulses or lentils. Okay, if you speak Hindi, dals, or if it is Tamil, it's parupu. Okay, varieties of these. These are 30% dry weight protein source. Okay, this is the primary source of protein. As long as you depend on them for your protein, you are in good shape. Okay, if you forage elsewhere for protein, then you will also get saturated fat and along with it troubles. So these are the main producers of the nitrogen containing compounds. So proteins have amino acids, and there you have nitrogen. So that's actually where we need the nitrogen. So these plants, here is the spelling for that word, they belong to Fabaceae. This is the botanical name of the family, Fabaceae, including this. And these are bulk of the crops that we grow, Fabaceae plants. And vegetables are primarily Solanaceae family. And then the starch comes from the gramineae, okay, the grass relatives like rice, wheat, barley, oat, etc. So before we begin on the Fabaceae plants and why they are important in nitrogen metabolism, etc., we need to remember these 4 bullets. They are really important regarding nitrogen metabolism. So the first point is the volume of matter, the bulk flow through these pathways that we are going to learn is really low compared to the bulk flow through carbohydrate and lipid metabolism. They are the main things. Bulk of the matter is in the form of carbon, hydrogen, oxygen without phosphorus or nitrogen. Okay, so those are the carbohydrates and lipids. And different amino acids and nucleotides, remember purine, pyrimidine, they have nitrogens. They are actually called nitrogenous bases. They must be made in correct ratios and at the right time. So if you want to make a protein with 300 amino acids, and out of the 20 amino acids, if you are deficient of only 1 amino acid, you're not going to make the 300 amino acid protein. Okay, let us say you have 19 amino acids and the missing amino acid is lysine. In translating an mRNA, if the third triplet is for lysine, and if you don't have lysine, that's it. You're not going to add the 4th, 5th, and so on. So it is all or none phenomena. So you need all the amino acids at the right ratio at the right time to make the right proteins. Same is the case with nucleotides. And a third point is amino acids and nucleotides are charged molecules. Okay, so these have phosphates attached and therefore they are significantly acidic molecule. And these have amino group and carboxyl group, both are charged molecules. And when you are playing with charged molecules, you can't have too much of them, then you will create electrochemical gradients. So therefore, this needs to be tightly regulated. And the nitrogen can be recovered and recycled, and as a result, the nitrogen compounds are scarce. The atmosphere has 70% nitrogen, but it is triple bonded, extremely stable structure, and therefore they are not readily available in the usable form in biological systems. So the biologically usable nitrogen is very scarce, and therefore the nitrogen metabolism is tightly regulated and very efficiently scavenged and recycled again and again. So this is to break the myth that you need to eat a lot of proteins. It's really not true. The amino groups can be scavenged and recycled by fixing into carbohydrates, and amino acids can be remade in our body. Okay, so you need proteins, but not a whole lot. So for these 4 reasons, the nitrogen metabolism is tightly regulated. Okay, because the flow is very less, and therefore you need to regulate correctly, appropriate things at the appropriate time. Similarly, correct ratios are important, and it is an all or none, and therefore again it has to be very tightly regulated to make the right things available at the right time. Then you don't want charge gradients there, and therefore again another reason why it should be regulated. And resource is very scarce, so you need to judiciously use. So these are the 4 reasons why this metabolism is lot more fine-tuned and regulated relative to the other 2, carbohydrates and lipids. They themselves are regulated as well. This is even more stringently regulated. That is all. So I'll stop here, and in the next class we will look at why leguminous plants are important and how the nitrogen triple bond is broken and fixed in the biological system.